I'm Dr. Deborah Winter. I'm an assistant professor in the Division of Rheumatology at Northwestern Medicine. I'm a computational immunologist, and I run a research lab that studies the gene regulatory networks of macrophages. I received a grant from the National Institute of Health to study the transcriptional regulation of macrophages in aging. I think macrophages get a bad reputation because we think of them as causing inflammation and inflammation being bad. But actually, macrophages exist in almost every organ of your body, and they're important for the health of the tissue. And they also are important in immune response because inflammation is good. When you hurt yourself or when you have a virus or an infection, you need inflammation to trigger your immune system, and then you want it to resolve. The issue is that sometimes things will go wrong, and you'll have chronic inflammation or you'll have defective wound healing, and this can lead to problems. The goal of our research is to understand how macrophages start to lose their function in aging. As you get older, they stop performing their functions, and part of this is because of the way they're programmed. So over time, the programming starts to malfunction. In our study, we're focusing specifically on macrophages in the joint, and we're asking how they change over time as you age, and what factors are involved in this aging. And we're trying to understand this at the genomic level so we can see how we can possibly in the future change this programming in order to increase the health of our macrophages and therefore the health of our joints. We're using high-throughput sequence technologies, which generate a lot of data, big data, if you will, and we're combining this with computational analysis of the genomic landscape to get a better idea of what the macrophages are doing in individual patients, and therefore be able to target our treatment for that specific patient. This is a precision medicine approach, and the idea is that we would sample the macrophages in a given patient and use this to inform clinical decision-making and have a more personalized approach to each patient. In another grant, which we will soon start, that was awarded by the National Institute of Health, we are studying transcriptional heterogeneity of macrophages in rheumatoid arthritis. We are using similar approaches, studying macrophages on the genomic level, but in this project we're focusing specifically on the role of macrophages in inflammatory arthritis. We will be using a mouse model of acute inflammatory arthritis to study how the macrophage subpopulations change from their healthy state when they preserve joint integrity to how things go wrong as inflammation increases leading to arthritis, and then how macrophages play a role in the resolution of inflammation. And we hope to be able to leverage this to understand how arthritis develops in patients and how we might help it to resolve. The final part of the study is to use patient samples. We obtained these through a consortium called the Reason Consortium, which is a group of over 6 institutions that perform minimally invasive ultrasound-guided synovial biopsy to obtain synovial tissue from the joints of rheumatoid arthritis patients, and we can use this tissue to isolate macrophages from the joint and study how the macrophages differ from one patient to the other. And in this study in particular, we're using these macrophages to look at patients at the peak when their disease flares, and then 6 weeks later as they hopefully respond to treatment. And by comparing the macrophage profile before and after treatment in the peak of inflammation and during resolution, we can determine how well our mouse model recapitulates human disease and whether we can use these principles that we're learning from the mouse model to apply to human disease. So it's very important to translate our findings using patient samples so we can see what kind of variability we observe across patients, and then we can actually harness this variability to better predict how different patients will respond to different treatments. Although genomics can seem like basic research far removed from the clinical arena of medicine, and the amount of data it generates can be overwhelming, I believe there's a lot of potential to translate functional genomics to inform clinical decision-making. At the end of the day, how the macrophages are programmed on the genomic level determines how they will function in disease and how they will respond to treatments. Understanding the gene regulatory of macrophages is critical for understanding disease and for determining how to treat patients.